So, I have given the title Learn Your Rat and Mice. So, whenever you do any preclinical studies, either of pharmacological or toxicological aspect, you should know about these species and their strains also. So, you should know about rat and mice. So, why and what particular things you should know, that we'll see. So, if we compare mice with humans, so there is say 85% genome similarity, whereas if we compare it with chimpanzees, there is 96% similarity. Now you tell me about rat. When we compare it with humans, how much percentage similarity is there? Genome similarity is important to know, because on that basis only you are close to the humans, so then you can determine your specific model. So you should also know about the number of chromosomes present in mice and rat. So in humans there are 23 pairs of chromosome, whereas in rat there are 21, and mice only 20 number of pair of chromosomes are present. So now you tell me why it is important for you to know the number of chromosomes present. Is it related with some genotoxicity studies or not? So one more thing you should know about rat and human brain. So brain structure, if you are doing neurological studies, so what are the differences? Actually you should know about all the organs. Is there any, like in liver there are differences. So if we see in brain, so cortex, human cortex is very much wrinkled, whereas the cortex of the rat is very smooth, and also the surface area. Now you tell me about surface area, what are the differences? So because of that humans are very intelligent. Next is about olfactory bulb. Rodents have relatively more evolved olfactory bulbs, so because they are mediating for their sense of smell than humans. So we know that rodents can smell from long distance anything. And now you tell me, there are any differences in cerebellum of rat and humans. So which and how it is better, and you just need to compare it. Or if I have left anything which is different in human and rat brains, so you can just note or let me know. Also, so we were talking about suppose rats, so you should know the differences in strains, because SD and Vista rats are most commonly used strains. So what are the differences between SD and Vista rats, and which studies you should prefer SD rat and which studies you should prefer Vista rats? So if we see the structural differences, then the ears of SD rats are short, and whereas Vista rats there are long ears. And if we see about tail length, SD rats have long tail as compared to Vista rats. So these are the observational differences. And if we see head, then SD rat is narrow head, and Vista rat has wider head. So these differences are just to identify the strain of the rat. Next is